The next part of the chapter focuses on measuring the electrical activity in the brain. The reason why we focus in language studies on EEG measures is because EEGs are very precise in matters of time. fMRI is very precise in locating the individual areas that are responsible for certain kinds of linguistic input, but EEG is much better at identifying exactly when certain things are being processed in the brain. So if you were to use EEG and fMRI together in a combined study, then that would give you both information about time and also about location. But sometimes a specific hypothesis might just be that we want to know about the timing of processing in the brain, and that's what we're going to look at next. So in order to understand the timing of electrical activity in the brain, we have to understand first why electrical activity can be measured in the brain in the first place. Where does electrical activity come from? So this is a neuron. We have about 100 billion of these filling our head. We also have other kinds of cells in our head that support neurons and the work that they do, about the same amount of other kinds of cells as we do of neurons. But these are the ones that carry information for us and allow us to do the kinds of computations that we do in order to live and be human beings. So here we have the beginning of the neuron. This is the cell body. These are the dendrites. These dendrites accept signals from other neurons. As you can see here, we can't understand a neuron in isolation. Neurons, the whole point of a neuron is that they communicate signals to other neurons. So here you can see the dendrites of a second neuron are receiving signals from this first neuron. We could also say that this neuron here is receiving signals from previous neurons that are speaking to it. Every neuron can have thousands of communications with other neurons. We're seeing here just one neuron communicating to a second neuron, but this neuron may be has the potential to speak with up to thousands of others on this end here. Now, what makes a neuron fire is a difference of electrically charged ions flowing into and out of the cell. Whenever a neuron receives signals from this neuron receives signals from this previous neuron, the way that those signals are communicated is chemically. This neuron here will release certain neurotransmitters or chemicals into the little space between the two neurons called the synapse. And in that space where the chemicals are released, the chemicals float across and bind with certain receptors on this postsynaptic neuron. There at that site, those chemicals will function like keys in a lock, and they will open up certain channels that will allow fluid to flow into the cell. And when that fluid flows into the cell, certain kinds of ions enter the cell, and they have either a positive or negative charge. And when that happens, this neuron will decide, based on whether they're positive or negatively charged, to fire or not to fire. And if the neuron makes the decision to fire, then a chain reaction will be sparked here at this part of the neuron that continues down the length of the axon until it arrives at these axon terminals. But all along the length of the axon here, there's going to be some shift in the polarity or in the electrical charge of the parts of that part of the neuron. It's going to become very positively charged compared to the fluid around it, and once the neuron stops firing, then it's going to be very negatively charged compared to the fluid around it. So basically what we're measuring when we measure electrical inputs in the brain is the flow of electrically charged ions in different areas. There's no actual electrical activity in the brain so much as there is just electrical shifts in the fluid movements that we can measure. Now it's important that we understand, because we have so many of these neurons throughout our brain, that we're not measuring the electrical activity of individual neurons. Rather, when we put an EEG cap on somebody's head, we're measuring thousands upon thousands upon thousands of neurons all at once, and their conglomerate or aggregated electrical activity. So in a study like this one that you read about in your text, we can present individuals with different stimuli, like words or non-words, with like pseudostrings that are characters that look like they might be letters, or icon strings that are actually pictures in a sequence, just like words would be letters in a sequence, and objects versus pseudo-objects. And when we present these various stimuli to individuals, we can measure their electrical activity in their brain at varying time points after the onset of this stimulus. The time point 0 is exactly the moment when the individual first saw the stimulus, like the word airplane. And throughout the course of the study, the participant will see lots and lots of words, or non-words, or pseudostrings, or icon strings. All these stimuli will be presented many times over and in different varying varieties. So they won't just see the word airplane, they'll see also lots of other kinds of words. But here's the moment at which those stimuli, whatever they were presented. And from that moment going forward, you can track how long it took before the brain was able to distinguish between different types of stimuli. So here, at this point right here, this is where we begin to see that the blue and orange lines split off from the other 3 lines, or from the other 4 lines. This orange and blue line are the objects and pseudo-objects. And you see that the brain doesn't really make a distinction between these objects and pseudo-objects until right about this point here, where the blue line splits off from the orange line. But here, importantly, what we want to focus on is it's about 95 milliseconds before the brain distinguishes between objects or pseudo-objects and other kinds of stimuli that could have the potential to become words. Down here you can see that the green line splits off from the other 3 lines. That's where we make the distinction between icon strings that look like this, that are like mostly pictures but in sequences like letters of words would be. And the other 3 kinds of stimuli, these 3 sets right here, continue on in their processing. Later we can see that the red line and the red line separates from the purple and the black lines here. In other words, the pseudo-font strings here are segregated in the mind at about, I don't know, looks like maybe 200 milliseconds. And then a very short time after, the pink line is separated from the black line, and this pink line represents these non-words. So we could make the conclusion that at about 200 milliseconds, these types of stimuli are being treated separately in the brain, that words and non-words are being differentially processed. However, our authors argue that while this is a safe conclusion, what's not a safe conclusion is to say that this is the point when we recognize the difference between real words and fake words. The reason why is because the sequence of letters that were used in this study here are very improbably connected. We don't see these kinds of letters in conjunction with each other very often in the real world. Like, how often do you see the letters S-I-U-P in combination with each other? I can't think of a single time when that appears in a word, but A-I-R would appear in lots of different kinds of words. So rather than concluding that this is the point at which we recognize words versus fake words, we have to merely conclude that these 2 types of stimuli are being processed differently. And in a follow-up study, one researcher found that we don't actually make the distinction between real words and words that look like they could be real words because their letters are being organized in a sequence that's more probable and more frequently observed in the natural world until about 350 milliseconds. And that's not really depicted on this graph because it was a follow-up study.